and very warm welcome to Iit Bombay and for this workshop on Big Data and Precision Medicine, which we abbreviated as BDPM 2020. So we are very pleased to see many of you here in person. We have been communicating to you from last several months. And idea for this workshop is essentially to create more awareness of this field on one hand, another to also enhance more collaboration, foster the joint activities together. So morning sessions are more formal when we are giving some lectures and making it more kind of basic educations. But afternoon we want to keep it really open so that people can interact, talk among the peers, and also try to establish more collaboration. Uh, we have combined two activities together for this particular event. When we have in the afternoon some participants who are for a course which is from continuing education program of IT Bombay, which is the CP course, where they're more from the PhD background kind of students, so they would like to have some hands-on training. So we will give them some training in the laboratory part, and rest of the delegates are essentially from the DST and UKERI funded project when we have the joint Indo-UK workshop, where we want the participants to really talk among each other and see what kind of joint work we can do. Maybe in the next 5 days, once we deliberate, we would like to come up with some sort of bigger activities of this consortium. So, with this brief introduction, let me welcome my co-convener and Professor Graham Ball. He has been really a great friend and great collaborator over the years, and we are very pleased to see him again back here in IT Bombay Campus. So, thank you, Graham, for being so generous for all of our activities and really supporting this initiative for big data precision medicine at IT Bombay. I'd like to hand over to Professor Graham Ball for a few words here.
So once again, thank you everybody for making the effort to come here. Thank you particularly for the UK delegates who've travelled some distance to get here as well. And also I would like to thank Sanjeeva for the warm welcome I received as ever. And it's a pleasure to be back in Mumbai again. So the purpose of this workshop is we're essentially at the beginning of a revolution. We have an opportunity through the generation of data, through digitization of biology, to start to understand diseases and biological problems at the fundamental level. So we've tried to gather here representatives both from India and the UK who have expertise in measuring things, profiling things, and then taking those things out into diagnostic devices. So one of the challenges with the digitization of biology is the complexity of the system. As a human being, we have around 35,000 genes, coding for around 200,000 proteins, bumping up to maybe 300,000-400,000 with post-translational modification. All of those can potentially interact with every other one. And then you consider that as happening at each cell, then and each organ, and there's a whole network interacting system that is then perturbed by disease. That is a big data problem. It's sort of on the scale of astronomical data in terms of its size. So the purpose really is, once we've measured the biological system to the best of our ability across the transcriptional pathway, then we need to do something with that data. We need to find things from that data that are valuable, that are robust, that can be utilized for diagnostic purposes, but also potentially for drug discovery purposes. Once we've done that, then we need to do something with those things. And then that's when we start entering through the clinical evaluation trials process. We start to develop point-of-care devices, because at the end of the day, we can't wheel a mass spectrometry very easily out into a community, although they're getting smaller and smaller. And we are able, as TAF has worked on, to take them to places like Mars, potentially, and able to profile things in very small spaces. And also we're able to sequence things. So things like minions, very small sequences, which can be taken out. But again, how do you deal in the community with that volume of data? So it's in some ways it's easier to take a small set of markers out. So the idea really is, firstly, as Professor Srivastava said, firstly we want to upskill people here to some extent, the young faculty, young students, to give them a skill set which they can take forward because they are the future. But also we want to build consortia that establish this pipeline from clinical sample through to diagnostic device, and to maximize the potential of all the steps along the way. So welcome everybody to the meeting, and I look forward to interesting lectures and fruitful discussions. Thank you.
Great. All right. Thank you so much, Graham, for the nice words. Now, what I will do, I will give you my first very short overview for the whole workshop, so that everybody is on the same page to know that what all we are planning for the next 5 days. And then I'll give you one detailed lecture, which will try to summarize a variety of technologies, which also we are going to discuss over the next 5 days. A few quick announcements before I start. So, some of you who joined a little late, we have the Wi-Fi in the room, which is the IITB guest Wi-Fi. I have shared the login and password on the WhatsApp groups. Also, we will just write the password here so that people can also join in if they need. In the lecture, you will not need much, but I think in the hands-on sessions, probably it will be a good idea to have your systems available and do connected with the Wi-Fi. Also, the lectures are going to be recorded. So in case you are asking some questions in between, take a minute time to make sure that your microphone is on and you are actually audible, not only to us but to the camera as well, so that then we can talk. So during the session, let's not have just random discussions. Please also turn on your microphone and then ask the questions. Thank you. All right. I'm sure small room I'm audible, but to the camera, is that okay, audio? All right. So let me start. I'm sure you are aware I'm the convener of this activity along with Mr. Graham Ball, Sanjeev Srivastava, professor in the Department of Biosciences Bioengineering at Iit Bombay. And this workshop, Big Data Precision Medicine, comes from the way that the entire world is actually moving ahead. With each individual is very unique, and we need very personalized medicine, personalized diagnostic for individual, right? That's the idea. And towards this goal, the classical biochemistry may not be sufficient. So therefore, we need a new biochemistry, which is based on the new tools, which is based on the omics based technologies, where you are really trying to investigate the things in the holistic approach. So that's where the big data is actually being generated right now, a lot from variety of technologies, from genomics, proteomics, metabolomics, many omic technologies, and all of them are contributing to the complex data set, big data, which is how best we can try to make knowledgeable sense out of that data is actually very important. And that's where then the artificial intelligence and machine learning comes into the play to really try to get the best out of the data. And that's where we thought to combine the two concepts of big data and precision medicine and try to first bring everybody to the same page about where the things are happening in this area. And then of course, we see how we can take the next action forward. So you are in Iit Bombay Campus. Probably many of you who are from India already are aware of Iit Bombay. Some of our guests from UK. Iit Bombay is one of the premier institutes of India, and we get the best of the minds from all over India. These are the best undergrad students, and we also have the Institute of Eminence status from our Ministry of Human Resource Development, Government of India. So we are well funded, and we do emphasize both on teaching and research at Iit Bombay, where we have initially mainly undergrad teaching was prominent, but now we have more and more even the postgrad emphasis about research. So we have almost 10,000 students on roll. We also have large number of postdoctoral fellows who are joining in, various project staffs, and there are 27 academic departments and research units we have. These numbers are slightly old, but still around less than 700 faculty members we have, and lot of adjunct faculty and visiting faculty. So my primary affiliations from Department of Biosciences Bioengineering, and some of you will be also doing your experiments in the department. So our department is focusing on lot of postgrad education and research, and we have essentially M.Tech Biomedical Engineering program. We also have Masters in Biotechnology program, and we have almost now 200 PhD students who are involved in doing the different type of research activities. And it's very interdisciplinary department when we have faculty strengths from the engineering to the medical to the basic science. So, all a spectrum we have, and that really kind of facilitate a lot of interdisciplinary research activities. So, please feel free to interact with people around, and many faculty and students who are working in different research areas. Today afternoon, in fact, for all the DST participants, we have planned to have a short visit of the campus tour and also the department and the facilities, whereas the students who have come to learn the technology, they should be doing the experiments. All right, so for our lab, which is essentially we are establishing proteomics facility, we have variety of instrumentation looking from the microarrays to multi-TOFs to different type of scanners and the fractionators to the QTOF. So Dr. Gandhi is here, she's handling the instrument at CRNTS. We have the Bayako T200 SPR and the microarray printer instruments, a triple quad mass spec. And more recently, we have established this national facility based on the high resolution mass spectrometry, which is now equipped with variety of instruments from Orbitrap Fusion, QXKTRIF, triple quadrupole, and also recently a next generation sequencer. These are generously funded by Department of Biotechnology, Ministry of Human Resource Development, and your Center of Excellence from Thermo Fisher Scientific. So about this workshop, I had given some idea, but let me just walk you through day-wise plan. So first of all, let me acknowledge our collaboration from Nottingham Trent University, UK, which has been over the years really strengthened further, and now we are looking forward for more joint PhD students, and in addition to the joint research activity, those will be actually really strengthening all the joint work which we are doing with Professor Graham Ball, and many of you are here from the NTU side. So we'll be actually looking forward for more active collaboration, and now actually things credential on paper when we have the joint co-supervision and the joint projects to go. Now for this particular activity, for today's event, I'm just flashing it through. You already have the details in the flyers and in your copies. So today I'll give you one lecture about which is summarize variety of technologies in the field of proteogenomics, and I just thought to also add you the flavor of other latest omics revolution which is happening in the field. After tea break, then Professor Graham is going to talk about various experimental strategies which are required, because generating data is very straightforward nowadays in the omics area, whether it's genome sequencer or mass spec, but how you plan to design that experiment becomes most crucial, because your data analysis eventually depends on that. And many times we actually always make mistake of doing experiment first, analyzing data, then we realizing, okay, we have missed out these controls, we have missed out these many replicates, and all those things becomes very, very critical to take back. So that's where I think this part was very important to think about, and he will also give you some idea for other statistical theory. Post lunch, we thought to keep it slightly, kind of variety of backgrounds of different applications for every day. So today we have Dr. Anirban Chatterjee. He will talk about some of the applications of genomics in mycobacterium tuberculosis patients care. Dr. Rashmi Rana will talk about the brain tumor proteomics based work. After 3 p.m. then we are going to split the groups into 2 groups. The DST participants, we will show you the campus around. Of course, you need to talk more among the groups, so we want to give you a little bit free time to interact more. And the CEP student, the participant for the course, for you we have the hands-on session for sample preparation, where we would like you to walk through several steps of if you have been given a tissue lysate or any kind of sample to start with, what kind of quality control checks you are going to do, and then also once you have got the proteins, how are you going to digest the protein for doing the mass spectrometry based experiment. So in the 5 days we would like you to continue your experiments every day. So this experiment will be overnight step, which again you will take forward for in solution digestion. The peptides will be ready for tomorrow to be injected in the mass spectrometers, right? So then you are going to start the experiments along with various theory which we will be talking in the afternoon session. Also, it's entirely up to you to make best use of the laboratory and the resources here to stay longer and do your experiment to the completion, because the timing is short for completing all the steps. So if you really wish to stay longer, you are welcome to spend more time in the lab. Tomorrow we are going to have again further session from Professor Graham, more about the data analysis statistical considerations. And then we have Dr. Ben Dickens. He will talk about the genomics role, talking about different disease context. Dr. Mike Johnson, probably he will talk about different devices that can be used for point of care diagnostics. And then post lunch we have Dr. Gokul Krishnan from NIMHANS. He will talk about epigenetics and metabolomics, how that can be utilized. And Dr. Firoz, he will talk about proteogenomics for ovarian cancer. Dr. Jayashree will talk about mycobacterium and how she has investigated that with different molecular biology technologies. So the afternoon sessions are giving you the flavor of different disease and different type of technologies currently being used to investigate different problems. And again, post that the participants who are for the DST workshop Indo UK, they will be staying in this room to discuss about some collaborative projects. We would also like to have more formal introduction for each one of us for the DST course that time, to give you the flavor of everybody what they are doing. And the CEP participant, they will move on to the lab again, to the BSB, to Proteomics lab, to now look for how to analyze those sample what they prepared, digested peptides, to run on the mass spec for doing label free quantification, and also will show you iTrack or TMT based label and how you can analyze that data. So these two activities will be planned for tomorrow. Then we move on to the 5th March, and Dr. Jayant Morgan is here, so he will talk about GCMS and metabolomics. Of course, he has miniaturized many mass specs, so he will probably give you the flavor of how the entire field of mass spectrometry is now progressing towards much a shorter version of mass spec, more clinically applicable. Dr. Mark Turner could not show up, so we'll fill in a session, maybe some hands-on session we'll do on the bioinformatics in this time. And Professor Graham will continue on the bioinformatics and big data analysis part. Dr. Neelam Shishit from ACTRAC, she will talk about how transcriptomics can be utilized for precision medicine, and she works on the medulloblastoma and various brain tumors. She will talk about that. Then tomorrow post lunch we have Dr. Asha Chandola from Dehradun. She will be talking about various type of decision making in the big data driven sciences. Dr. Suman Thakur is coming from CCMB Hyderabad. He will talk about how different type of mass spectrometry technology that are advancing right now. And Dr. Arup, he will talk about one of his recent experiments on MS based quantitative proteomics for the brain based work. And then we would like to have the Indo UK participant to talk about potential joint grant opportunities. I think there are already some calls out there. Can we make the best use of the interaction and the synergy what we have together here? And then we are going to have a session for the metabolomics for the hands-on for the participants, more on the untargeted metabolomics, which is global metabolomics. And again, timing will be short, but we will show you a couple of runs of the different metabolomics experiment and also the data analysis in between time. So the 6th March is going to be quite busy because morning will be same way, the way every day we are doing the session, but afternoon we are also going to have a dedicated brainstorming roundtable discussion. So in the morning we have Professor Rahul Pawar. He works on the immunotherapy aspect. So he will give you flavor about when you take out the potential targets, how you can now convert them to the therapeutic strategies, right? And he is using CAR T cell therapy and other new antigen based vaccine therapies. So he's going to talk about those things tomorrow. Dr. Shrikant and Mahesh Kulkarni, they're both mass spectrometry experts. They will talk about the clinical applications of mass spec as well as PTM based investigation using mass spectrometers. And then we will have Dr. Amit Dutt. He is the expert of genomics and translational research. He'll give you different examples. Dr. Jingde will give you the much broader perspective of proteomics field, from where it started and where it's going to lead. So as I mentioned, this day we are going to have a little bit more busy schedule. So this evening we will have a roundtable discussion, which we have not put in detail here right now, but will be a lot more people joining in that afternoon. And idea will be to develop more policy decision making kind of opportunities, and we'll have some of the officials from the British High Commission also will be joining for that roundtable discussion. So the Indo-UK participants and some other clinicians, industry leaders will be joining in for that meeting. But rest of the CP students should continue back to the lab work, and they should do the targeted proteomics which now will be utilizing a triple quad mass spec to identify, you have identified some sort of target from either proteomics or metabolomics. You can now validate using targeted based experiments. We will actually show you some peptide validation using this workflow. Finally, last day we will again have more talks from Mr. Rahul Purwar. He will talk about immunotherapy aspects. Mr. Graham Ball will talk about oncology, how different tools are available for research. Dr. Gauri, she comes from the industry background. She will talk about various diagnostic opportunities based on different proteins and other targets which they are identifying and their role in precision medicine. And I'll try to summarize with different other applications how these fields are evolving. Finally, for the Indo-UK participants, it will be a little bit free time. We want now to summarize all your activities, not here, but maybe while we are having a sightseeing somewhere in Mumbai. And the CP participants can get a flavor of the genomics technology and some experiment of genomics, which actually we would like to start little bit on 6th itself, a day before the library preparation, which takes overnight, and then 7th we'll do the data analysis part. So for all the CP participants, the students essentially who have come from variety of different institutes and different places with a lot of expectation and hope to learn new things. Of course, you are very serious. That's why you have come to this event. But we would like you to also take this course very seriously, because we will have quiz every day for you. Not for the Indo-UK participants, don't want to scare all of you, but only for the CP students, you will have the quiz every day, and that will be online quiz. So each day when you will start in your session at BSBE, you will be given some questions from the previous day, whatever you have learned. So go back, read some material, clarify the doubts, and then and there is no penalty if you don't do well, but if you do well, you will have some prizes for that. So I'm sure you are now happy with this. All right, so let me also thank our funding partners, and especially UK IERI, UK India Education Research Initiative, Department of Science Technology, Iit Bombay and NTU for generously supporting all our activities, and then CP office for facilitating this course, and various industry partners who are helping in different ways of technology contribution, different application support, and different type of reagents which are required for the course. So thanks to all of them for supporting all our activities very generously. So finally, we welcome all of you, and we really look forward to have new partnerships, new collaborations, new projects, new funding opportunities together. And this is not going to be just a conference and workshop. You listen the lectures and go back. We would like to actually come up together as a team and really show that we have achieved something out of this particular 5 days. And probably we have written some proposal. We have also submitted some grants and things like that. So we are very much looking forward for much more informal discussion and much more stronger bonding after these 5 days. Thank you so much. Any questions for the workshop? Any comments, anything which is not meeting your requirements, not meeting your expectations? How would you like to see this course evolving these 5 days? How best we can help you better? I think if you have any comments, please do let us know now. Later on, you can only write the feedback, it was good or bad, but right now, you have an opportunity and we can improvise, right? So,
the lectures that people are giving, the idea is to in part showcase what people are doing, what their expertise is. So we've got a very interdisciplinary team here, and if somebody has some idea or interested in saying, well, what if we try this? That's the key thing. If we get one question that says, what if we try this? Then this workshop will be a success.
absolutely. And please make it interactive, because it is not a formal conference, right? We have kept the numbers very strict, very low, just so that we can enhance more interaction. We talk more among each other. So don't feel afraid. It is not like a classroom or not like conference. Just make it more interactive.
Everybody can speak, no matter how
No,
you are.
right? Absolutely.
Good idea. Don't just come from professors. Anybody else?
No? No, so I will, we have a lecture now and then tea.
Okay.
All right, so let me now start the formal lecture, right? on the SWAYAM portal, so that it can be widely made available to a large number of audience who would like to learn, who cannot be physically here. So, type of proteogenomic technologies are evolving, and eventually what we see currently is a very revolutionary phase when every day you are hearing something big is happening and very different, which was not thought of earlier, right. So, biology was moving of course at a very steady pace, but only after 2000 or so we started seeing the things at very different scale, and now we see the things at very unprecedented scale. So, I really feel excited and fortunate to be in this day and age when we are part of these revolutions which are happening in front of us, when we have no idea that how best we can handle this kind of huge amount of information, right. So, I will talk to you about little bit different type of proteogenomics technology. So, that is my first part, and second some of the revolutions which are happening, which I think is lot of revolutions in this area which is happening, and good idea for us to be on page that which is all things happening around in different part of the world. So, let me start first with a broad overview slide, which probably gives an idea that the central dogma of life, which is very complex. It starts from DNA, RNA to mRNA, protein and metabolites. All of these information, all of these molecules, they are all inside our body, work together, right. But many times when we work on one technology, like we only work on genomics or only proteomics, we think we have understood the system very well, but that may not be reality, because all of this works together inside the system, right. So, for any physiology to be defined, we need to have a holistic view, we need to have the systems level information together, whereas what we are looking at only snapshot at one of these molecules. And if that is the case, that we have to look at only one of the molecule, then of course, first you have to create a baseline, and that is where the DNA based information and genomics comes into the play, when you would like to first establish the reference for that individual, their sequences, something normal, most of them normal, some abnormalities, all of that you need to document, and then how many of those have really made the functional RNA molecules, and based on that information, then how many of those actually translated to the proteins. Now, proteins being very dynamic functional molecules, they are the one used by almost all the pharma sector as the therapeutic candidate targets. So, if you want to develop the drugs, mostly people are not looking at DNA or RNA, they are actually essentially looking at proteins, because to control protein, then you can actually try to control disease much more effectively. So, therefore, proteins becomes much more important, while both of these molecules are equally important, RNA and protein, but you see more action happening at the protein level. Therefore, lot of therapeutic strategies are developing towards proteins. Finally, the metabolites is of course much more important, because these are the ultimate molecules which are reflecting based on your metabolism, and therefore, if you can measure the metabolites, that can be clinically highly relevant. So, now you can see that there are layers of information, which is each of them are their own significance, but also we have to prioritize which one could use in a given context. Nevertheless, the genomics have really become very robust, very strong, very reproducible, very much achievable. Therefore, it has really spread very widely. Large number of people have started using genomics in their own research. Transcriptomics also I think made significant progress over the years with microarray, real-time PCR, followed by now the NGS RNA sequencing based approaches. It has also become quite robust. Then come the proteins and proteomics, which is very dynamic, and just the nature of the proteins are so complex, so dynamic, We are not handling now 4 base pairs, we are handling 20 amino acids, so many side chain chemistries, lot of complexity. And in the DNA based technology, you have very robust methods like polymerase chain reaction, which can amplify your DNA, and now you can get tons of DNA for doing your experiment. In proteins, you don't have the analogous PCR machine yet. You have to keep purifying, keep extracting, then only you can get enough protein to do your experiment. And each time when you store a protein for 1 week, you are making a second batch, first batch is degrading, first batch is losing activity, and then you are again bound to go back and think about comparing your two. You are always into that race that okay, how fast, how quickly I can do these experiments. And so therefore, we have still not reached to the maturity enough when we can say that now we have a instrument from which we can amplify the protein on the day which we want to do our experiment, and we are very robust now to take it forward. So, and then comes the data analysis, because again like genomics, because it is slightly advancing, there are a lot of tools which are available, although I am sure NGS is still facing the problem of data analysis in bioinformatics, but in general the technology is still saturated enough now to really have the robust bioinformatics platform. In the field of proteomics, still there are different type of workflows involved. You think about you want to compare the proteins from one to other differential level, and now you have too many methodologies available. You can use 2D gel based method, you can use microarray based method, you can use mass spectrometry based methods, you can use other label free platforms. Within mass spectrometry, you can have label based methods, label free methods. Label based methods are again different types, iTrack, TMTs, SILAC, variety of ways. And label free, again we have no idea how to quantify the signals because they are all label free. So, the field is still evolving and really getting challenged with the enormous complexity of the proteome and how best to robust manner we can analyze the data. So, therefore, this becomes very important molecule, but it's still very challenging to really reproducibly achieve the things again and again. Therefore, lot of SOPs requirements are very, very crucial in this field. Coming to metabolomics, very easy to perform the experiments, but the data analysis is very challenging because the databases are not very well annotated yet. Even for human, which is the of course most look after, everybody has a lot of investment in that area. Still, we don't have the good libraries and databases for all the human metabolites. So, when you want to confirm this is a given metabolite, you need to have MSMS confirmation for the spectra, which you have for only limited number of compounds. So, while this could be very clinically revolutionary in nature, but because of the way the databases are limited and data analysis is complex, it has not reached to its full potential. Otherwise, it can be very easily executed to the clinical settings. So, I hope you got to feel that each one of these omic technologies have their own pros and cons. They do add a lot of value to the biological question which you want to address, but they also offer a lot of challenges how best you can get the meaningful information from these technologies. So, therefore, you have to choose your questions and accordingly the right type of technologies which is best platform for your system. If of course, your lab is having availability for multiple platforms, you would like to integrate as much information as possible, so that now you can try to get much more holistic information, you can integrate the information. One thing which is going to be much more powerful, that you cannot do so many experiments on the wet lab side, right. If you can generate good hypothesis from at least limited number of experiments, and that data could be fed into some good models, can you now predict for unknowns, and that's where the lot of computational algorithms will be in the play for looking at systems type of network analysis, right. So, all of this is possible if we are really going beyond our bias, beyond our comfort zone, beyond looking at only one part of the picture at a time and claiming everything out of that. So, that is our biggest mistake, that we think we have learned everything just by looking at one snapshot of any of these molecules. So, let me now go back and take you for the longer journey of starting from genomics field and moving ahead. So, of course, this is the Sanger sequencing, which all of you are aware that how looking at ATGC sequences at some point was challenging, right. And idea was that if you are looking to sequence a DNA template, you are adding the primers, DNA polymerase, deoxyribonucleotides. In addition to that, you are also adding dideoxyribonucleotides, which are fluorescently tagged, and fluorescently tagged with either DDATP, DDCTP, DDTTP or GTP. So, now each one of these nucleotides are fluorescently labeled with different colors, and during the reaction, whenever this one of the DDNTP, which is dideoxyribonucleotides, it binds over there, it is going to terminate that reaction, because it is there is no 3' hydroxyl group over there to continue that chain. And that is where now you can get the fluorescent read when this particular nucleotide is passing. You can look for that color change and you can denote what that nucleotide is, right. So, therefore, now you can get the GCAT sequences very easily. This was the classical way of doing this whole work. Then of course, this led to the big success, which was for the Human Genome Project, which took many years, almost by doing this kind of shotgun sequencing method. It took almost 15 years to first time decipher the draft human genome maps, which led into this papers of Nature and Science in 2002, 2003. And that is the first time which we learned that we have now idea for all the human genes. Numbers were still not very clear. Numbers are still keep defining and redefining and refining further. And right now we think there is estimate of 20,300 genes in human, which is not huge number and not very different than many other organisms what we know. So, therefore, genome is not very different in human versus other model organism which we are looking after. So, of course, there are many other molecules which are beyond that which I just talked to you, including RNA, proteins, metabolites. This is what makes systems much more unique and different, right. So, the Human Genome Project was of course one of the biggest success in the omics field, which led us a path that now we can understand a system in its totality, first time looking at all the genes of a human being, and a project which can be done with the scientists in a very time bound manner from different countries, different teams together, and all with a deadline of one common goal, right. But over the years, then this technology has really reached to much more maturity, and we have now got the next generation sequencers. So, second and third generation sequencers, which are all now aiming towards looking at a very quick way of doing the sequencing. I'll just shut off the.
This model shows a typical nanopore made from.
So sorry. No, not working.
The house extract.
Yes. All right. So, I don't want to show a lot of videos in terms of the with the commentary, but I just want to flash it through to give you the flavor.
Designs
So,
and manufactures bespoke nanopore structures for a range of applications. In nature, nanopores form holes in membranes. In Oxford Nanopore's system, the nanopore is inserted into a membrane created by a synthetic.
is there audio from there?
Video will be carried by the HDMI.
So, I turned off the audio from my system, but it's still. Okay, let me try it the other way around. All right, sorry about that. So, let me start again. So, is that not connected? You turned off something from there, right? Fine. Okay, so welcome back. So, we are talking about different type of next generation sequencing technologies, right? And as I mentioned, there are different type of principles, different platforms which are currently being used. I am not going to talk all of those in detail, but I am just keep flashing this video, which is based on the nanopore technology. And likewise, what you will realize that there are different type of advancements are happening currently, where again the engineering disciplines have contributed a lot when they are trying to really make the sequencing robust, much faster and much cheaper. So, the goal is we can now do the sequencing maybe in a couple of hours time and in much the lower price as compared to what we used to do earlier. And that has really contributed heavily for this whole field of genomics. So, what you are seeing here that the same principle what we saw in the Sanger sequencing when you are looking at differences in nucleotides and trying to do the fluorescence based labels. Now, you are looking at their conductance changes for each nucleotide and you are trying to measure that very rapidly and how fast you can now measure those variations and then get the sequence. So, there are different type of principles which are involved and different vendors are currently in the market who are making very robust instruments, but I am not going to give you detail for all the sequencing platform, but just to give you the idea that in last 18 or 20 years post human genome project, still many technologies have advanced in the field of genomics, which has made genomics very robust. Otherwise, the shotgun proteomics, shotgun genomics to say, shotgun genomics and conventional approach was not very straightforward for every lab to do the whole genome sequencing. But now it is very much reality and lot of big projects are happening in the field. Additionally, people have also looked at the gene expression analysis using the microarray based platforms, where idea was to identify the network of genes and look at the information at the genome wide scale. Again, that was something which gave us an idea all the molecules of life at once, right. And this technology again enjoyed a very good time, especially from 1990s to I would say 2005. That was the time when many genome projects were getting completed. We had idea for many of the EST projects, lot of SNP information, and people were doing lot of microarray based assays to identify the biological problems of their interest. So, again this you can generate lot of data with a very simplified manner. When you are having two population, let us say cancer cell and normal cell, you are extracting RNA from those, labeling them with Psi5 and Psi3, mixing them together, hybridizing them on the chip and looking at now the expression of these genes and trying to look for the which color you can see based on your control, your treatment, or there is no change or the empty spot. So, accordingly then now you can normalize and subtract the signal and look for the expression changes, right. So, both microarrays and of course, definitely the sequences, they have really contributed a lot, and how this information has really helped and changed the perspective and changed our perception of clinics. Something like this has happened when breast cancer was thought to be as one of the disease, which is one disease of course for the women suffering from breast cancer. But after looking at their gene expression profiles, what they realized that all individuals are not same and they are all actually showing the different type of gene expression. And eventually when they try to analyze the data, they could actually make it 4 different subgroups of the breast cancer. One is luminal A type, luminal B type, HER2 and basal like. And what they realized that some of these women are actually having the estrogen receptors and progesterone receptors high level, HER2 was negative. This luminal B also estrogen receptor and progesterone receptor were positive, HER2 was negative, but estrogen were also quite low in this case. HER2 was only having the higher level of Herceptin, and in case of basal like this is all triple negative, this is no receptors available. So, of course, drugs which are going to work on these individuals also based on the which receptor they can bind, and therefore, the same drugs are not going to work exactly same manner in all of these women. So, this was the first time when looking at gene expression profile, people are able to decipher that we need to now profile each type of cancer or each type of disease to look at their molecular subtypes. So, this sometime if you think about now why despite years of advancement a disease like cancer is still so challenging, because we treat them many times as one disease and we go ahead with the type of treatment which is very standard chemotherapy and set treatment given, and we do not have ability still very robustly to try to analyze those things at much more detailed level. And that is where this workshop and kind of our idea to look into precision medicine, looking at each individual at the personalized level becomes much more important. So, this was the long back PNS paper and the study based on conventional methods, but I hope this gives you the flavor that why we are thinking about to really dissect out the molecules and investigate them at much deeper level. So, let me show you one of this particular news article, which is shown for one of the Hollywood film star Angelina Jolie, and the BRCA mutation now we know is very important for the breast cancer screening, but earlier people had no idea that why they should screen for a specific gene. the standard set of mammography and the standard clinical tests are sufficient for doing this kind of diagnostic. But if you want to know the disease early, disease ahead of time, then of course only molecular signatures can give you the idea, right? So the awareness for this thing came very much with this particular incident which happened with Angelina Jolie. In her family there was a history of ovarian cancer and breast cancer from her mother and aunt, and she was of course at the very high risk level of these diseases. So she thought to go ahead and do this type of gene testing ahead of time in California. This was not a routine thing done that time, and of course then you see what happened in the clip.
Her face is known around the world, and now her secret is known too. Angelina Jolie revealed in a New York Times editorial Tuesday that she had a preventive double mastectomy earlier this year, after discovering she has a gene defect that greatly increases her chances of getting breast and ovarian cancer. She writes, "My doctors estimated that I had an 87% risk of breast cancer and a 50% risk of ovarian cancer, although the risk is different in the case of each woman." While Jolie's genetic risk was very high, cancer experts say it's also fairly rare.
So 5% of breast cancers in the United States occur in women who have the genetic mutation, the BRCA1 or the BRCA2 gene.
Doctors say women with a family history of breast or ovarian cancer at an early age are more likely to carry the mutation, and insurance companies usually pay for genetic testing. For those who test positive, Dr. Rachel Brem says having both breasts removed like Jolie did is a very personal and difficult decision. It should be made with the help of genetics and cancer specialists.
It doesn't mean that that's the right decision for everyone. And the important thing to do is to have frank discussions.
Jolie said she went public because she wanted women in her shoes to know they have options and can still look and feel like a woman after a mastectomy.
Well, I think one of the reasons that we're seeing an increase in what we call bilateral or a mastectomy on both sides is that there are better reconstruction techniques.
The actress was treated in a California breast cancer center.
We hope that the awareness she is raising around the world will save countless lives.
Jolie's star power is already drawing attention to a complicated choice some women must face. Kelly Daschle, Associated Press.
All right, so you have seen that people started using it, and of course she raised a lot of awareness that people should go for genetic tests beforehand. And these things have now become quite routine for even locally. If you go to Mumbai Tata Memorial Hospital, they will do BRCA testing. Of course, in Europe and North America, it has become one of the standard tests already. So the basic biology, looking at the new technologies and bringing to the clinical test, I think it's all getting now reality. And that's where the precision medicine is really moving ahead with the advancement of all of these knowledge what we have generated over the years' time. So other example comes here, which is Dr. Lucas Wortman. So he is a researcher at University of Washington, working on the leukemia, and unfortunately he himself got leukemia. And what he realized that he was given the chemotherapy and he was not responding well. And at that time in US, precision medicine was not approved from the Congress. So it was not in their standard healthcare what can be covered. But his university allowed him to go for the sequencing of his genome, and looking at his genome, then what they realized that he is suffering from some very unique mutations which were not thought of in leukemia. And based on those mutations, then if you now can give targeted therapies, then response could be very different. Please watch this clip.
Fortunately, during my last year of fellowship, started feeling a little bit more run down. It happened more suddenly this time. This past July was diagnosed again with relapsed leukemia. Was admitted into the hospital and started on very strong chemotherapy. Unfortunately, though, the leukemia didn't go into remission after being treated with this very high-dose chemotherapy. There were no real promising treatments on the horizon, and so things looked rather bleak. But at the same time, as I was being treated, my leukemia genome was being sequenced by the Genome Institute here at Washington University School of Medicine. We got back the sequencing results, which showed that there were promising targets that weren't even in the scope of our standard treatment protocols for this type of leukemia. because of those results, I was able to start less aggressive chemotherapy that I could tolerate, and then targeted therapy that had been approved for actually renal cell or kidney cancer. Within a few days, my blood counts improved, and within a few weeks, my leukemia was in remission. So it was really going from a situation that looked really bleak to now a situation where there was hope again.
All right, very interesting example, because in this case, he would have never thought of to take a drug which is known for renal cancer, but because he was suffering from this mutation, which is FMS-like tyrosine 3 kinase mutation, and there is already known drug for that, for a different cancer treatment. So idea is, if you now know the target, that which is the having the most impact for that disease, you can look for which are all approved drug from the FDA or other kind of regulatory bodies, and then try to utilize that. You need not to have that it already prescribed for that particular disease, and then only is going to work on. So that was the pretty much case here, that when he took sunitinib, which is known for the renal cell cancer, he actually showed very positive response, and the tumor really got suppressed from that. So I hope I'm able to convey you the need for precision medicine, need for looking at the targets, and how these kind of knowledge which we are generating has really started making impact, right? So as I mentioned, the genomics have become quite robust, not only from technology point of view, but also became much more affordable, right? And probably you have seen some of this graph in other lectures, when people are able to now showcase that how best you can do the genome sequencing at much lower cost, which has really kind of exceeded whatever one could expect even from the Moore's law, that in such a short span you have seen such a rapid advancement. Technology has become robust, results are very high quality, and now the costs have really gone down. And as a result, now if you can sequence your genome in $1,000, 70,000 rupees, in just 5-6 hours time, then I think you have now much more detailed wealth of information for that individual, rather than looking for certain tests which you are only looking at superficially. So you I'm sure are already reading a lot of articles in this field, and every day something is happening. UK is pretty big in lot of big genome projects, lot of big papers have already published, big initiatives are happening. Recently, this you might have seen in Nature for the Genome Asia 100K project. They revealed the diversity of the Asian population, lot of new information which came out from this. So from the Human Genome Project, which happened in 2002, 2003, we are in 2020. It's still not too big a change, but now we see the scale is very different. We are not talking about any more one individual, we are talking about 100,000 individuals, and we are looking at the data not in the 15 or 20 years time, we are looking at data in at least couple of months time, right. So this thing is paradigm shift. As I said, I feel very privileged to be in this day and age when the revolution is really happening in front of us, and we see how rapidly this whole field is evolving. So from the Human Genome Project to almost reaching to the personal genome level, right. And of course, we are looking at now whether everybody should have their own genome sequences available. Maybe in 10 years later, if we meet for another workshop, everybody will have their own CD-ROM, their own databases, their own genome sequences already available. And when you are going to see a doctor, you are not going with a blood report, not urine report anymore. You are going with your entire omics data set, right? And doctor is going to put on their computer, looking at the entire sequences variability. And now you talk about what your symptoms are right now. Can we now profile your immediate metabolome, immediate proteome, and now try to map those together and see where the changes are happening, right? So you are being compared against yourself. You are not being compared against the big population and the preconceived knowledge that we have, right? So that's the big change which is happening, and is going to really rapidly advance our knowledge. So what is next? Of course, we have to think about how best some of these knowledge, these technologies could really translate to the clinical settings. It's already happening very rapidly, and I'm sure in next 10 years probably it will also have very much reach to your nearby local hospitals. So some little bit basics about genome and proteome comparison. We have talked about genome, we have seen the rapid advancement. And of course, I'm more advocate of the proteome, right? So I'll have to tell you why proteome is better and different. So when we say genome, we are looking at all the genes of that particular individual, and proteome, we are looking at all the proteins of that individual which is present in organism. The context could be different when we are looking at a specific cell like mitochondria proteome. It means all the proteins of mitochondria. We are looking at a specific organelle or a specific sample type. We are looking at blood proteome, for example, plasma proteome. So, all the proteins present in that particular sample type. All the time we are not only seeing the whole organism proteome or whole organism genome. Of course, you are only going to do at whole genome level. Proteome will be organ specific or cell specific as well. So, now these are two interesting examples. If you look at the classical textbooks, the caterpillar, how it is doing developmental different stages of transformation from the pupa to the larva stage to the adult, and finally the beautiful butterfly, right. All of this looks so different. If you are trying to follow the developmental stages of this particular organism, you find everything is so different, but the genome of them is all same at all of these stages, but the proteome is what makes them so different. So, the proteins are keep changing as they are developing. So, this actually shows you the need to look at the dynamic molecule, how they can make the individual different, while they are all same from the gene point of view, but they are all very different from the protein and dynamic molecules point of view. Take another example of frog. If you think about the tadpole larva and the adult frog, these two represent, of course, the evolutionary biology you are all have taken the textbooks to see that how amphibians, they show so much changes for metamorphosis to really change from their the living in the water bodies and moving on to the land, which everything in the physiology gets changed. So, they go through again the complex cycle of the development stage, which is all same at the genome level, but the proteome is dynamic and keep changing, and this is what makes this particular tadpole larva which is living in the water to make it really now go and open air and survive on the land. So, you need much different functional molecule to provide that immunity. So, I hope this gives you the flavor that while both are equally important, genomics and proteomics, but genomics gives you the fingerprint of the life, gives you the information blueprint, but the proteome gives you at each snapshot, each dynamic level, the changes happening at that particular level. So, if you want to characterize this pupa at this stage, you have to look at the proteome at that stage. You cannot compare that with this stage, because these two are very different. So, now if you are looking at somebody's disease, you have to look for that individual, their proteome that time. You cannot just say, okay, the genome was for this individual belongs to this category, and I can only translate information from that at any time, because things are very dynamic, right. So, we are taking two classical example of caterpillar from butterfly to convert that, which shows that how genome is static and proteome is dynamic, and they are of course going to really dictate the phenotype of any organism and of course the complex physiological system. So, therefore, what we have to really look into the generating the blueprint from the genome level and also understanding the complex dynamic molecule, both proteome and metabolome in that way can be equally powerful. And the changes which happens, of course, why we see those variations, again the refreshing about the basics. When from the we are doing the central dogma of life, from the transcription and translations are happening to make the protein. So, at the alternative splicing level, the RNA can convert into different type of mature mRNA forms while exchanging the different exons, and therefore, the same gene could give rise to different type of mature mRNA, and further each of this protein could get modified with the different type of residues, different type of molecules, and could be very different functionally. For example, addition of just a phosphate makes it phosphorylated, which can be involved in lot of signal transduction activities. Glucose molecule can make it more glycosylation, methylation, hydroxylation. So, alternative splicing and the post transition modification are the two major events which makes your static genome very dynamic and very different at the proteome level, right. And that is where we need to finally look at actually things at this level if we want to look at certain actionable target for certain therapeutic aspects. So, proteome in that way looks for a very broad overview of characterizing a given individual at the protein level, to look for their activities at the protein modification level, where proteins are localized, and also these proteins do not work in isolation, they do interact with the different proteins of the similar kind of functions. So, then which proteins are interacting with those other proteins, and together we can now define the function of the different protein molecules. So, all of these things are while they look very simple, but to actually understand them, our recent technologies or even latest technologies are not yet matured enough to really do these things in a kind of yes no manner. We can say okay yes, but maybe because these are interacting, but maybe there are maybe more interactors, right. There is a modification happening at the phosphorylation level, but may also be glycosylation is happening. So, we do not have very clear 0 1 kind of matrix or yes no answer that this is the only thing happening, right, and that is still the big challenge which is fundamental in front of us. So, to really understand the proteome even at the cell level, one single cell if you want to characterize, you are looking at different type of activities happening, how the proteins are making complexes, different type of interactions happening not only from protein to protein, protein DNA, protein lipid interaction, protein carbohydrate, sugar molecule, and of course, different type of PTMs getting changed, protein activities are getting altered as a result of that. So, what we are talking about a very complex set of information which is how to capture that in a very robust manner, and therefore, proteomics despite we are now 18 years later than genomics is still, because of all these inherent challenges involved, we do not have the very robust set of one or a few technologies which can answer all of these questions. So, therefore, you are always limited with variety of workflows, variety of technologies, and for which we do not have too many experts actually who can make those kind of maniacs book which can teach you from the cloning from the step 1 to last step, and you can follow your all the protocol and every lab can follow that. So, that is where we are still very much challenged, right, and challenges are actually so real that even my lab which has around 20 people work, and even till last week we keep deliberating how to analyze the data set for the complex differential proteomics, how to make it really reproducible. So, that 20 people if we give the same data, can they generate same output, and very challenging truly speaking, because everybody makes their own recipe, they add little bit salt, some flavor, some sugar, some this data set, this database, this software, and at the end same data is not going to give you the same set of significant proteins, right, very challenging. So, that is where I think our problems are enormous and our workflows are not robust enough to give a same answer, and that is where we have to really think very closely how best we can overcome these challenges. So, proteomics, while we are talking right now which looks very relevant, but it has really evolved over the years from many years, and it started in 1970s with different type of activity which was happening in the field of electrophoresis, and scientists like Close and Farrell, they made 2-dimensional gels which separates protein based on their molecular weight and the isoelectric point, and then different advancements are happening in mass spectrometry, but only when the soft ionization techniques like electrospray ionization and MALDI, when they came into the picture, then mass spec could start to be used for proteomics based application. Additionally, we were also having a lot of sequencing activities happening from the EST and nucleotide sequencing, which toward the genome sequencing, all of them were getting matured, and only in 1995, the very first time the term of proteome being coined with Mark Wilkinson from Australia. And over the years, then we started following the success and trail of genomics, and only 2014 and 15 first time we got the map of the human proteome, draft human proteome map. And map here, the aim was if we have 20,300 or so genes, for each one of these genes there should be a protein, at least one protein made, right. Although from my basic slide which I showed you, you would expect many proteins to be made with alternative splicing and post-translational modification. So, the numbers cannot be exact, numbers could be a million, right, because how many proteins we can have, but at least there should be 20,300 proteins, right. So, that many protein sequences should be available for us to detect experimentally, and that was the goal, very straightforward goal given by genomics team to the proteomics, that can you now provide experimental evidence for every gene what we have, is the protein already being formed from that or not. And so far we have no information for all of these proteins at the experimental level, while these two papers have been published in almost 5 years ago now, which showed that time 17,500 protein evidence. So, they showed that at least 17,500 proteins you can detect their sequences in the mass spectrometry. This was the paper with 2 different scientist groups, one from Johns Hopkins IOB together, one from Kruster's group from Germany. So, 2 of them in the same issue showed that mass spectrometry can provide evidences of these proteins at the peptide level, but it is still not full genome to be sequenced. And other group in 2015 showed that antibody could be used against the proteins, and showed that you can use the different type of expression based analysis, localization of the proteins to confirm that these proteins are actually expressing. So, two different approaches, one in nature, one in science showed, but still none of them could show that all the proteins what are expected we are able to provide evidence for them. That is why they are draft human proteome reference map. So, if we have 20,300 total genes to look for and we have still only 18,000 proteins for which we have evidence. So, we have 2,300 almost which are missing, right, for which we have no idea where they are in the body, or we are not able to go with the detection limit from our instruments. Instruments are not able to capture those. These are so fast, so dynamic that we are not able to, our sensitivity or the scan rate of the instrument is not fast enough for them. They are so low abundant. They are only stage specific. They only express only in a specific type of developmental stage. We have no idea. This is known as missing proteome. A full field is developing right now, which is aiming towards giving the at least identifying more and more protein, providing evidence for that, right. So, again you can see while it is a challenging field, but so much opportunity that it still has not matured enough like genomics. When you say okay, now we have already a genome reference map and everybody can be sequenced. Even for one human we do not have the full proteome information, and still there are 2,000 or so proteins which are still missing, right. So, a lot of gaps still in the field, lot of opportunities also for all of you, because the proteome is very dynamic. We have so many modifications, so many networks happening, and again as I mentioned, if we really want to look at the comprehensive characterization of any biological processes, we have to limit our bias, our mindsets, and our technology per se, for which we are very much comfortable. We should look at all of them together and look for then how dynamic proteome could be characterized with our various technologies available. So, there are different technologies which are currently being used in the field of proteomics. Of course, I cannot talk much about all of them right now, but I will at least give you some flavor of some of them. But as I mentioned that if your goals were so complex to look for enzyme activities, to PTM modification, to protein localization, to protein-protein interactions, so many wide questions you had, you cannot have that one technique which can answer all of these questions. So, you need arsenal of technologies, and each one of those you have so many variants already available. So, things from the gel based technologies like 2D gels and dye gels to the gel free technologies, variety of microarray platforms, variety of mass spectrometry based advancement, all of them together are at least trying to unravel some of this problem, trying to provide some answer for these kind of challenging questions. So, one of the best way which has come forward is a shotgun proteomics, which has learnt a lot from the 2D gel based proteomics which was happening earlier, when people used to take your two different samples from let us say control and treatment, separate them on two different gels, and then you are going to look for all the spots on the gel and try to excise the spot, identify them in the mass spec. But when you want to identify in the mass spec, you realize that the protein which is present in that tiny spot is very little amount, and when you are doing peptide extraction from that, you are not able to get sufficient peptides for mass spec to read, and therefore, you have identified interesting protein, but you have no idea what this protein is. The name of the protein is not possible. So, people then realize, can we now take our same complex sample which we were separating on the gels and directly now separate in the mass spec, and that became shotgun proteomics. Initial concept of this came from a technique known as mud pit, which is multi-dimensional protein identification technology. Again, for CP participant, please take notes whenever required and take ask questions about what is not clear. So, mud pit was one of the approach which showed that like 2D, which involves two different principles of IEF and molecular weight from SDS page, mud pit could utilize different type of chromatographic separation, and without running the gel you can separate the complex proteome. So, that is the first technology, and then the concept of shotgun proteomics came forward from that. If you have a complex cell or tissue samples, you are lysing the cells, you are then let us say putting them in the urea buffer, you are having reduction alkylation steps, digest them with the enzymes like trypsin or chymotrypsin or lyse, and then running the mass spec to generate either MS1 spectra or MSMS spectra or MSNS spectra and analyze the data set from that. So, based on this information, now one could extrapolate that and extend that further to now do the label based and label free proteomics, because now you want to compare the states, right. Like in the 2D gels you were running on two different gels and you wanted to compare control versus disease. Now, here without running the gel, can you now compare control versus disease just looking at their spectral count, how many spectra of a given peptide you see in control, and for the same protein and same peptide you see in the disease state. So, I will just walk you through for this particular concept here. This is the label based quantitative proteomics, which is currently being used as a gold standard in the field of quantitative proteomics. If you want to compare your healthy versus disease or control versus treatment, if you can use any of this technology like iTrack or TMT, you can publish something very I would not say robustly, but it can be straightforward peer review process. You will not have much trouble in terms of showing that what proteins you have identified, those 4 changes, and that kind of expression is actually quite robust. So, there are 2 terms which I mentioned. One is isobaric tagging for relative and absolute quantification, or iTrack. Other is TMT or tandem mass tag. Both of them are N-terminal sequence based labeling strategy. So, once you have digested your protein to peptide forms, those peptides you want to have a generic strategy, so that now their N-terminal could be bound with these tags, which are the TMT or iTrack tags. So, now you got a very generic strategy. I am not talking about too many other things which are possible, but these 2, as I mentioned, these are now currently industry standard of doing quantitative proteomics. You have many other options of doing that as well, but these are very robust strategies which works very well for the straightforward N-terminal based sequence based labeling. Now, let us say you are doing one experiment from a given organism when you are looking at 4 different developmental stages. Let us take our metamorphosis example of caterpillar to the different pupa larva stages, right, to the butterfly. So, now you want to characterize proteome of one organism at different developmental stages. You are taking some samples from 4 of those stages, and now you want to look for which are all proteins changing dynamically in 4 conditions. Or you are doing a cell line based experiment, you are giving a drug, and after every 3 days you are taking out a small aliquot of that, and you want to see which are the proteins getting changed because of the drug which you are giving, how many proteins are inhibiting, how many proteins are not changing based on the response. So, now you got 4 conditions to be compared, and you got a tag which, to simplify that, let us give a number. So, these are iTrack 114, 115, 116, 117, 4 different tags being used. And what you want when you are thinking about any labeling strategy and you are comparing 4 different states, you do not want the labeling to be different in 4 different conditions, right. So, you want your these 4 developmental stages not to get affected because your labeling was different in 4 stages, but you just want to see the protein changes, right. So, therefore, you do not want any molecule which is added on the protein should be different in different molecular weight. So, now in this case you are having these 4 different tags, but you are adding a balancer region, so that the entire mass of that remains 145. So, you are not making any changes in overall proteome. Yeah.
In this case, do you also quantitate the proteins, like suppose you are taking at 4 stages, so the amount of the protein intake initially should be quantitated before labeling or no?
Absolutely. The question is that when you are doing this kind of quantitative proteomics work, are you going to start with the same amount of protein and do you quantify that, right?
Yeah.
You definitely should do that. In fact, I will give you some more detail about it. So, let us say if you are doing this experiment when you are starting with 100 milligram of tissue from 4 conditions. So, first you would like to start with same amount of tissue as well, then you again quantify that you got same amount of protein, and now let us say you are digesting 200 microgram protein from all the 4 conditions, but no, that is not sufficient. What we have learned, and others have also learned in the field, that protein based quantification not sufficient, because what you are doing labeling, you are doing labeling at a peptide level. So, you should do quantification at peptide level, because a lot changes from the protein to peptide when you are digesting, and that actually is a huge change, right. And if you are only under assumption that my protein that I started with will remain 200 microgram on, even after all my steps, that is not correct assumption. So, we do now peptide quantification, and again some of the lab sessions people will learn the skill that why you should look at those kind of quality control checks. So, only if you now quantify peptides before labeling, that will be the most accurate quantification for your 4 different conditions to be compared. So, peptide level quantification is most accurate for this kind of quantitative strategies. So, once you do that, now let us say these tags which I am talking, they are going to add a balancer region, so that each of this iTrack becomes 145. So, you are not adding anything different in 4 different conditions, everything is got 145, but you got these unique tags which at the MSMS level, when you are doing the MSMS and you want to fragment your ions. So, with the collision induced dissociation CID or HCD, when you dissociate that, then you can separate those fragments, and now that is going to reveal your quantitative changes. So, now you can see that for a given peptide, what happened to that peptide in my 4 different developmental stages. and I am going to use that information to quantify my signals and look for the fold changes of the protein across 4 different conditions. Also, because I am doing the MSMS, I already have the sequence of that peptide. So, now I can know that what this peptide is from databases and what this peptide belongs to which protein. Now, think about also the comparison with the gel based techniques. Of course, you can do a lot of experiment on the gel based work, but even if you have a good protein, you may not be able to identify that. In this case, anything which is significant, you already have the sequence available. So, you can now quantify them as well as you can identify the protein. So, that is what is known as isobaric tag for relative and absolute quantification. You can get the idea for quantitative value as well as you can identify the protein. Now, at the end of that information, now you can compare your different stages, you can make the heat maps, you can look for what happened to my development stage 1, 2, 3 and 4. Of course, things were not very different in the initial stage of development, but probably things became much more dynamic as it went along further. So, then this gives you much more dynamic picture about each of the 4 conditions which you wanted to compare. This is what is happening very much standard workflow in the field of quantitative proteomics, differential proteomics, which if you want to compare different states. So, some of you I think have also mentioned that you are keen into iTrack and this kind of workflow. We do a lot of iTrack and TMTs, and of course, we can give you more detail about those. In this case, we are looking at some of the brain tumor samples, patients suffering from healthy individual, no disease, but says grade 1, 2 and 3 of the tumor, and then each one of those we extract the protein, digest peptides, label the peptides individually for each of the patients. Now, you can mix them all together after doing labeling. Now, you can do the fractionation either using the isoelectric focusing based method or chromatographic based methods, and then you can run on the mass spec to generate the spectra for these MSMS at the reporter ion level. So, this is what people do in the quantitative proteomics using iTrack. Other advancement which is happening in the field is label free quantitative proteomics, which is now gaining much more popularity, and of course, there is the less cost involved in doing the experiment. You do not have to buy very costly proprietary reagents of iTrack and TMT. But also the information can be very powerful, because if you think about a clinical study which you are doing, which is always challenged with the number of sample which you have. So, this month you have only 20 sample, and next month you got another 30 sample, and following month you got another 20 sample. If you have to do an iTrack experiment, you should ideally wait for all the 100 sample to come over the last 4 or 5 months time. Then only you can make some proper reference pools, and now you can think about your labeling strategy, how best you can label your 4-plex or 8-plex or 10-plex accordingly, but you have to wait. So, now some sample were stored in your freezers for those 6 months time, and some were stored for only 1 month time. You have already done quantitative proteomics to start with, right? Without even starting your experiment, you have now variability introduced to your sample because of the storage conditions. Now, if you have to think about how reproducibly you can do, how reliably you can do, label free can be very powerful. You get a sample today, and let us say within 7 days time, if you make a strategy that I will do protein extraction, peptide, digestion, and immediately run in the mass spec, generate the library for that particular patient and store that somewhere. So, now likewise, whenever I get a next sample, I am going to do the same process in a week time, then all the time your variability is within 1 week time for that individual. And what you are doing, you are generating patient wise, each individual wise, a database for their peptides. Then this information could eventually be used to compare across. So, at the end of 3 months, now you got 20 healthy and 20 disease, and end of 6 months you got now 100 healthy and 100 disease. So, your n can be keep changing and keep increasing, and you can reanalyze the data. Now, many times you have lot of information for your diseases. You have different type of clinical features. You got some genomic mutation information. So, now you can reanalyze them in much straightforward manner with the label free data, which you cannot do with the iTrack type of data, because that is already you have mixed, you have pooled, and you have got some very specific labeling strategies over there. In this case, you can reanalyze data, mix and match as and when you are moving along with the new information. So, here what you are doing the same process. Let us say we are taking plasma sample for this example. You are doing depletion of the plasma to remove the abundant proteins. You are doing the protein extraction, reduction, alkylation, digestion. All of these are almost same processes. Then running in the mass spectrometer, and then what you are trying to look for the spectral count, how many peptides are eluted out from the chromatography. So, in this case, what we are very careful that our chromatographic aspect is very reproducible. So, if a given peptide is showing me elution at 25 minutes, at the same retention time, the same peptide should come whether it was in the healthy or disease state, and how many spectra of that we can monitor at that retention time. So, you give very small window of error for the looking for this kind of analysis, and therefore your internal standard, your proper standard for data normalization, and your IRT peptide which can look for your chromatographic shifts, all of that becomes very crucial here, because you are relying that on a given time a healthy is showing, let us say, 2 spectra of peptide and disease is showing 10 spectra of that peptide. So, to be really sure that it is the same peptide which we are looking at, you need to have lot of other quality control checks in place. But if that can be done, this is a very powerful way, because you are not modifying the protein from external chemicals on one hand, which will always have some impact with the iTrack or TMTs, and you are not buying the costly reagents, you are not limited with the doing experiment at the same time only, you can do as or when you are moving ahead with your experiments. So, there are different strategies currently going on in this field. I will not talk about how the DIA and other technologies are evolving, but idea is that you can do quantitative proteomics using label based or label free manner. Other technologies which are getting more robust is targeted proteomics, which also you will get some flavor in the course as we go along. Idea is you have identified a set of peptides which are showing the best differential expression. So, this peptide is showing me very high fold change from my healthy to disease condition. So, now let us narrow down and for the time being remove our bias at the protein level and narrow it down to the peptide level. And what you will be really surprised, I am sure we will have more time to talk, interact more. We have now started looking at our own data, not at protein level, but also dissect out peptide level. You see how many peptides are reproducibly seen across all the patients. And what you will be surprised now, for a given protein, let us say there are 20 peptides you can measure in the mass spec, but only maybe 5 of them are actually seen across all the patients. Now, these 5 are the ones which are the most robust and best bet for you to now take forward, because if you make any reagents against these 5 peptides, whether you raise an antibody or you make a targeted assay, these are going to work for these peptides. If you were to look at broadly that protein which could generate in silico 100 peptides, you are working on certain regions which are not going to be common across all the patients. So, now if you can know that which are the peptides which are most reproducible across your conditions, then you can only monitor those peptides in the triple quadrupole based mass spectrometer, or you can also measure them in the Orbitrap based configuration. One of them is known as multiple reaction monitoring, other is known as parallel reaction monitoring. Idea here is you already know, you have narrowed down the peptides you have discovered from the discovery work. Now, you are only focusing on those. You tell mass spec only select this particular peptide, rest everything goes garbage. So, now only that peptide you are fragmenting further in the triple quad, you are looking at the retention time and monitoring the different transitions to look for what intensity of that peptide we have. And at least you should have 3 peptides to be really confident that this protein is showing the very high fold change from the as compared to disease, and then only you can make those assays. Parallel reaction monitoring is slightly more ambitious concept. Here idea is for every peptide which is running in the mass spec, we are generating their transitions and their quantification directly. And of course, SWAT is the next in that manner, where for each of those whatever peptide you are acquiring, you are trying to generate their quantitative values for the targeted approach as well. So, all of these are reality, but when it comes to the getting more comfortable, of course, this one is much more straightforward, probably much more tightly controlled. So, if you want to bring to the clinical assays, the SRM based assays, selected reaction monitoring based assays, are the currently most powerful to really have the translational value. So, here let us say what we have done so far in our comparison, which was, let us say we are talking about control cell disease, we narrowed down a protein which is looks like potential biomarker to us, and we have for that protein we got 5 peptides which are very common across all the patients. Now, for these 5 peptides you have now measured their level in this particular experiment of targeted proteomics, and what you found that only 3 peptides are actually showing very good response and good intensity across all these patients. Now, of course, you have increased your n of the patient, you have added more numbers, more unknowns, and still these 3 peptides are holding true. For these 3 peptides, now you can generate synthetic peptide. You have the sequence for amino acids, you can generate synthetic peptide, you can now label them heavy with heavy label. Only now run this synthetic peptide in the mass spectrometer, generate the MSMS spectrum for that, and now you run your unknown sample and see where you overlay the spectra. So, now you can be very accurate in terms of quantification of that peptide. So, now you can say I have developed an assay for which this particular patient should have this much value for the quantification. This is what you see all the time from ELISA and other kind of assay which doctors give you, that the range is this for this particular test, right. So, can we come up with that kind of range for this peptide? And these things are happening now at the very fast pace, and these things are getting reality. And even based on one peptide, there is the first FDA test was approved for the thyroid cancer when they try to measure thyroglobulin, And just because it was so reproducibly seen in all the patients, just from one peptide they could generate this serum assay, which is now FDA approved and used for the thyroid cancer screening. So this part was really revolutionary in many ways and became Nature Method of the Year in 2013, in fact, 2012 to 2013. Based on target proteomics, how you can define a specific target and you can try to monitor the transitions for this. And now you don't have need to look for the antibodies, because earlier for any validation experiment you have to go for ELISA, Western blot, different other technologies. But now you are not limited with any of those. You can simply look for the peptide sequences, develop these kind of serum assays, and you can be confident to take it to the next level. Of course, then we need the miniaturized mass specs if you really want to deploy this in the clinical settings. But of course, if you are very happy with these leads, you can develop some different type of tests and different rapidly diagnostic tests for these kind of diseases. So mass spectrometry has definitely contributed a lot heavily. It's still not to the perfection level, but a lot towards making these goals looking much more reachable. And whether we look into now proteomics or even metabolomics could also be easily done using mass spectrometry based approaches. Instrument remains same. The workflows are slightly different. Proteomics involves lot of sample preparation part, which makes it much more complex, because each sample brings its own inherent challenge. You think about in this room, I'm sure people are working on variety of sample type. If you look into the plasma serum type of sample, you are going to be challenged with different type of abundant proteins. If you are looking at, let's say, cerebrospinal fluid, you have a lot of salts, so much low abundant proteins. You are looking at a plant sample, which could be a leaf, has a lot of chlorophyll. You have the root sample, lot of phenol. You are working on any other organism, has many other interfering components, lot of salt, lot of other interferences. So again, you need to know your system in which you are working on, and need to know the sample preparation strategy very much robustly first of all. I think that's one of the major challenge why people think that proteomics should deliver very robustly, but they don't see the results very easily, because sample preparation is very much creative part, and you need to know your biochemistry very well to really make good samples. And if your samples are good, I think technologies have really become quite robust, like NGS, which can give you reproducible result, but you need to make your samples ready and with the same SOP all the time. So this part is the most creative part, and still I think is most challenging part, whether we do discovery proteomics or targeted proteomics for the sample preparation strategies. But if you think about metabolomics, the sample preparation is very straightforward. You are simply doing some organic solvent extraction of your metabolites, and therefore you can employ some generic strategies of metabolomics, which can be very robust. But the data analysis part is still remains the challenge, which as I mentioned, because of the limitation of the databases available. I don't want to make all of you biased only for the mass spectrometers, because there is a lot more value for other technologies as well, especially protein microarrays, which in a very miniaturized form, it can give you so much information. So if you have thousands of proteins printed on the chip, and you have very small amount of sample available, whether it's a clinical sample or you have a drug which you want to screen for, in a very, very small volume, you can screen for thousands of protein on a miniaturized scale. So you are thinking about now a biochemistry of the thousands of molecule at very, very rapid pace with a very small volume. So what I am talking about is protein microarrays, which is limited in the nature, because there are not many companies which are offering you the total arrays for human or any other organism. So you are limited with only few companies or few collaborators who are working in this area. That's the major limitation. But otherwise, technology is very powerful, because you are looking at now proteome wide expression, and you are looking at the things at a very, very small volume. Now, if I just want to give you the comparison, so that you don't feel that I'm just advocating unnecessarily for microarray, think about your plasma sample, right? Plasma sample, if you take crude plasma, run in the mass spec, what you will see? Only highly abundant proteins, only immunoglobulins, only albumins, and abundant protein. None of the biomarker which you think a biomarker, you will see that in the mass spec, until unless you move out all the abundant proteins for the sample preparation part, which will also going to remove lot of low abundant proteins. When you do the array based platform, array based screening, you don't have to do depletion, because dynamic range is much higher for the readouts here. So now with the same crude plasma or serum, you are screening with some dilution of course, but you are now without any sample preparation strategy, you are now running on the whole chip, and you can now get the answer. So you are less doing the sample preparation part and looking at much more dynamic range of the overall protein changes of the low abundant proteins. So this is where you can think about pros and cons. If the goal was to look at biomarker screening for certain targets, I think these kind of platforms could be very robust. If the goal was to look for the new targets to identify, of course, then mass spec could be one of the valuable technology to start for the screening strategies. So, protein microarrays are also, while is not so popular right now because of the limitation of the chip itself, but have been used for variety of applications, from variety of protein-protein interaction, protein small molecule interactions, and different type of modification strategies. But whenever you use any technology, you have to always remember that the reactions has to mimic the biochemistry. None of the proteomics can just mimic the entire biochemistry what you want to do. So, if you want to do phosphorylation on a chip, you have to think about to add ATP, the kinase buffers, the right condition of incubation, everything, then only the kinization will work for that phosphorylation activity to happen. And likewise, you have to provide the conditions for glycosylation and other type of modification, then only you can identify them very robustly. But a lot of these things are happening, especially from the blood based samples. The biomarker screening is one of the big area for the protein microarray based applications. Once you have identified many of these targets, you would like to now look for which drug could bind and inhibit these proteins, which is going to create problem. So, you are looking at protein-protein and protein small molecular interactions. There are different type of label free biosensors available. One of them I am showing you here is the SPR, known as surface plasmon resonance. In this case, you have, let us say, the proteins of interest for which you have an antibody, if immobilized on the gold chip, and you are now passing your analytes, and after binding it shows you this kind of binding curve, which is known as sensorgram, which shows your how much on rate and off rate it can have for a given binding to either a small molecule or protein which is binding to that. So, this is one of the example, but there are many technologies like bilayer interferometry, different type of other platform which can be used for doing protein-protein and protein small molecule interaction studies. So, I could only finish my one part, but let me almost stop here with this slide to again re-emphasize what I started with the central dogma. Now, let us think about central dogma in the physiological system is now we are doing it with technologies, right. But let us not forget that we are still looking for central dogma for the complete information. So, we have to look at layer wise information from genes to transcripts to proteins to metabolites and maybe finally the phenotype and phenome information. But can we integrate that? We cannot look at the things in the isolation and individually. If we integrate that along with other clinical features which is available from the other information, then probably we could make much more meaningful information, much more educated sense of that data. Otherwise, we are looking at only superficial layers, and that is where probably despite so much years of advancement and lot of technology, we are still not able to make that impact, because we are only looking at layers and not together information. So, just to summarize this, I hope I gave you flavor from the genomics age, how things have really rapidly evolved, and still we need to look at the dynamic molecules like proteins, and I try to give you some flavor moving away from the gels to the mass spectrometers and some of the technology which can be used for interaction analysis. All of them are equally important, and I am sure during your work you would have used one of them for certain things, and you would also want to use others for other applications, other questions to be addressed. And at least good to be aware of what are all options available for your activities, for your research, so that you can really choose the right platform for right questions to be asked. And one thing which we at least definitely like in our lab is essentially do not get biased with the technologies and platforms, because all of these technologies are available to us, so we want to use them only to address the right question of interest. I will stop here. I will take this thing in maybe another time when we have time to talk more about it. But idea was to give everybody some flavor to get started, that where you are going to get data, so much data, and that data is not so straightforward information which you can just simply decode easily. So, you need lot of specialized eyes to think about, starting from the experiment what you started with, the technology platform, the data output, everything you have to be really knowing the in and out of that. Then only you can make good sense of the information, and that is what Professor Graham-Ball is going to give you the further detailed look about experimental design of that. Thank you so much for being so quiet and listening to lecture. But I want some questions from the students now. Please ask. Don't feel shy. Do ask questions, and we are going to ask a question for sure, right, tomorrow. So, you can ask today. Something is not clear, something, or if you think something you have already known about it and you want me to answer that, what things are happening new in that field, these are all kind of valid discussions. So, feel free to make it interactive.
I say to my students, if you now have to go and explain this to somebody else, what do you need to know?
Absolutely, other way around, yeah. Yes.
TMT more? Like how
Right.
it can
So
work?
the only names are different. One is tandem mass tag, TMT. Other is iTrack isobaric tagging for relative and absolute quantification. Companies are different. iTrack sold from SciEx and TMT from Thermo Fisher. The concept remains same. They are both competed for the same knowledge that the peptides could be labeled at the N-terminal sequence with the same chemistry same, but they only change the reporter ions. Like in iTrack you have 114 to 117, they have different numbers for the labeling, and they are always in the competition with the how many plex you can do analysis. So first iTrack started with 4 plex and then became 8 plex. Thermo came with a 6 plex, then Thermo brought this 10 plex. iTrack now probably brought 12 plex, and now Thermo has got 15 plex. So it's that the race is happening. Idea is who can capture the market, because the kits are proprietary item, lot of value you have to pay for buying the reagents, but the concept of the labeling remains the same. But what is more interesting here, which I have not talked, all this concept of the labeling came from one of the technology which was the ICAT, which is isotope coded affinity tagging, which was very similar to the 2D and DIGE labeling. So the how the science evolves is very nice. I did not talk about the story of that. The DIGE labeling started from the Psi3, Psi5 labeling, right? Looking at a specific amino acid based labeling. Then people thought, can we use the same concept for mass spectrometry based labeling? And they applied the ICAT kind of concept for the same cysteine residue based labeling, and they only made the heavy and light form of the ICAT 2 plex. Then eventually that translate information to the 4 plex and eventually multiplexing information. But that's not only one change like a multiplexing, but what is more important, going from the amino acid based labeling, which was in the DIGE case and ICAT, now technologies have become mature to look for the whole N-terminal based sequencing or N-terminal based labeling strategy. So these are the kind of major changes, and technology has really evolved in that manner. Yes.
So, you mentioned about array technology, protein array technologies. Do you know if anybody's ever done mass spectrometry of the bound element of the protein array? Because there might be things bound to the protein that are bound to the protein.
That's right.
Yeah.
So looking for potential interactors from microarray or even from the SPR for that matter is another interesting area. Microarray, if you think about, you have a glass slide or nitrocellular membrane, and then you are saturating the array surface with a blocker initially, because you don't want the unbound areas to give the signal. You are blocking with the super block or BSA and this kind of milk material. So they make the whole thing very dirty. So now doing mass spec from that makes it very difficult. So then that has not yielded a lot of success. But then people thought we are also doing the protein-protein interactions on SPR, which is the gold very clean slide, and over there we are not blocking with the super block or milk. So we have simply PBS washing and those kind of things are good to go over there. So then that has been used now, and when we have done experiments to integrate SPR with the mass spectrometers. So for example, if you are doing binding of antibody printed and one protein is flowing over there and this particular protein is binding. So that you already know, this is for X and Y interaction already established. But what about unknown things, right? So now you have an antibody, but you are not flowing a protein, but you are flowing a complex lysate. And from that lysate you want to see which are all complex materials binding to this antibody of interest. So but the overall binding what is happening is very minimal. That is not going to give sufficient peptides. So if you can currently, if you are using Bayacode system, which is the 4 plex, or earlier Biodata system for the 6 plex. Now in this system, if you are using on all the 4 channels, same antibody and same reaction if you are doing, now you are able to enrich that population to get more amount of protein binding to that protein of interest. So then you are now able to elute them out, and we do this thing in multiple cycles. So let's say 30-40 cycle, if you are doing the same experiment, you are stripping off whatever is getting bound, and then you are generating sufficient pool of the protein for digestion. Then probably you have sufficient peptides, which we have now able to do the mass spec on that. So this actually, of course, in the line of what you are asking, this is an interesting area which people are looking for unknown interactors, because otherwise you are always bound with you already know that these X and Y binds. You have the reagents for one and you want to confirm that. But what if you have only one partner and you want to identify the unknown partners? So can now you look for unknown interactors and characterize them using mass spectrometer to know what those proteins are binding. So if you can do on gold slide, it's much cleaner surface, more compatible for mass spec experiment eventually. Then probably you can do the experiment, but amount of analyte bound is very minimal. So you have to enrich them in multiple cycles. You keep eluting them out, make sufficient 30-40 cycles of that. Then only you have protein for digestion and analyzing the mass spectrometers. Yes.
First of all, I would like to thank you for such an informative talk.
Thank you.
And starting from basics to the very advanced one, and it's very nice talk.
Thank you.
And my question is, suppose we identify a drug target, suppose a peptide or anything, we tag it, and we know that there are several compounds from natural sources, natural extracts, which are used for treatment in our traditional medicines like Ayurveda, etc. They are treating many medicines, they are using many extracts and they are doing it. So can we just bind them, interact them, and use them, wash them and use it, means identify those compounds or alkaloids, whatever they are, from these mass spec based techniques, instead of going for code based SPRs?
Right. So, the mass spec can be high throughput in terms of giving you idea, but it cannot be confirmatory totally to give you the exact detail. And that's where you want to narrow down, from the larger to the smaller numbers. And once you really want to go to drug discovery pipeline, you have to come to Biacore kind of platform, because you want to really be sure not only a binding is happening, but what is the strength of binding and how long the binding stays for the on rate, how quickly it goes off, or how long it stays for the off rate. So then only the drug based kinetics could be developed. So that is definitely the end goal to go.
Initial screening.
Initial screening can be done in the mass spec.
So just following on from that point, you consider something like tissue microarray, which is a tissue sample bound with splice. If you could consider applying maybe a TCM or IOL compound to that, washing off,
Right.
and then doing the mass spectrometry on the cells that are there with and without the compound, you could actually then potentially identify what is bound and what is not bound to the cells.
I think very nice, good experimental plan for you. So, I think he just combined many different good concepts. Tissue microarray or reverse phase protein array is already in play right now. So, in the hospital, the doctors do not have access to very sophisticated technologies, but what they can take, they will take the tissue lysate, simply print those on the miniaturized platform, and now they look for abundant proteins biomarker. Let's say for the PSA you are looking at CEA protein, and you just simply want to look at what is the expression of that particular molecule in your given sample. So then tissue arrays are being used for that purpose very heavily. Now, what Professor Graham Ball has mentioned to you, that if you are preparing same kind of strategy, but now you are treating with different drugs, and now you are taking each one of those and now preparing a samples from that further for mass spec analysis, that could be very powerful amalgamation of two different technology platforms. So, I think this is what we want to see. Also, when I get more time to talk to you, we will see that how best one could integrate the multiple technologies and make good use of them together, because each technology have their own strengths, but also like when we are talking about arrays separately or SPR separately or mass spec separately, how we can integrate some of these to try to get some new information which is not possible individually, right? All right, I am sure we have a lot to discuss over the days. My team is keep showing me the stop signal, and we are getting late. So please do join for the tea break outside, and then I think we will be back within 5 minutes time for the next lecture. Thank you so much.